Can the current state of cancer treatment be improved? Is there a need for more selective therapies? The answers to these questions have led researchers down an interesting path, exploring the potential of a molecule known as AOH 1996. AOH 1996 is a small molecule PCNA inhibitor that selectively targets and kills cancer cells. This molecule was identified through a rational drug design approach, specifically aimed at targeting transcription replication conflicts. These conflicts are a major source of endogenous DNA double-stranded breaks and genomic instability, which are key factors in the development of cancer. Proliferating cell nuclear antigen, or PCNA, is critical to DNA replication and repair processes. AOH 1996 enhances the interaction between PCNA and the largest subunit of RNA polymerase II, known as RPB1. This action results in the dissociation of PCNA from actively transcribed chromatin regions, inducing DNA double-stranded breaks in a transcription-dependent manner. Interestingly, researchers found that a point mutation in RPB1's PCNA binding region can confer resistance to AOH 1996. This discovery provides important insights into the mechanism of action of AOH 1996 and how cancer cells may potentially develop resistance to this molecule. One of the most promising aspects of AOH 1996 is its ability to suppress tumor growth. This molecule can be orally administered and is metabolically stable, making it a practical and effective option for cancer treatment. Whether used as a monotherapy or in combination with other treatments, AOH 1996 has shown promising results in suppressing tumor growth. Even more impressively, it achieves these results with no discernible side effects. The implications of AOH 1996 for cancer treatment are potentially game-changing. It provides a new and unique therapeutic avenue for exploiting a cancer-selective vulnerability. By targeting transcription replication conflicts, AOH 1996 may offer a more selective and effective approach to cancer treatment. In conclusion, the potential of AOH 1996 is vast. Its unique mechanism of action, coupled with its effectiveness and lack of side effects, make it a promising candidate for future cancer therapies. The journey to improving cancer treatment is a long one, but with molecules like AOH 1996, we are moving in the right direction.